Hello everyone and welcome to today's video. So this is the second lecture video for chapter 5. Remember, chapter 5 is on the extensions and modifications of the basic principles of inheritance. So last video we talked about single locus extensions, or last lecture video that is, we talked about single locus extensions, and in this video we are going into the sex related extensions of inheritance. So remember, we have talked about sex linkage already. So sex linkage, when you have, a gene on the X or the Y chromosome, let's say, just A or little a here, so this individual will be heterozygous. So that's a gene linked on the X chromosome. Remember, then a male would be, normal with that genotype right there, but then, possess it with this phenotype right here because it's X linked. The male is hemizygous for this and would express that phenotype. But recall, this is all from chapter 4 if you do need to review yourself on that, but that is a sex-related extension of inheritance. So now we're going to go into some other weird ones we come across in genetics today. As you can see, there's a big goat down here coming up in this picture. Goats are a good example of what's known as sex influence. So exactly what it says, the sex influences the outcome. So characters express differently in two sexes. The expression, of course, is influenced by sex. So for male goats, so males and females are different here when it comes to the beards. So the beards are the key here on goats. So for males, let's make up some letters here. So the hierarchy of how this works. So if a male is big B with a little b, it has a beard. So say this was a male here. This male could be this. That could be the genotypes. And then B plus is beardless. So as you can see, B plus, that means it's kind of the wild type. But if a male, let's say, so assuming this is a male goat right here, so this would have a beard. And then also this would be bearded as well. Let me write that a little bit better there. This would be bearded as well. So both of these would have a beard because this is what matters. Now, if this male goat was B plus, it would then be beardless. So that would be a beardless goat, male goat. So now females, it's the opposite that occurs. So for a female, a B plus is beardless. So same thing here, but it's a different hierarchy. So the B plus is greater than the bearded. So a female, if we're looking at the female list over here of what could have a beard, the only genotype that could have a beard is heterozygous recessive right here. If this goat, so it's like this would be this female goat over here. So for beardless, it would just run the normal way. Oops, little b. I keep writing that one backwards, or the homozygous version. So all of these, remember, we drew a little table here, a made-up table. So these would be the bearded ones here, and these would be the beardless ones here, and this would be the sex up here. So interesting how the sex influences the orders of the hierarchy of these genes. It's almost making an allelic series, but it's not, a full allelic series. It depends on the sex, depends on the series and what's dominant and what's recessive. All right, so that's sex influence. Next one here is sex limitation. You see some roosters and a chicken. So the phenotype is only expressed in one sex, but it is autosomally encoded. So similar thing up here as sex influence. However, it's autosomally encoded. And it's only expressed in one sex. So it's limited to one sex, which is what that means. So on roosters, we're looking at the amount of feathering on the back here. So this is something that's autosomally encoded and only in, male roosters. And the amount of feathering is higher. So this, let's say this was a male rooster right here. This male could either be, homozygous or heterozygous and would show a lot of feathering. If this male was homozygous recessive, it wouldn't have this feathering back here. Now, on the other hand, if we look at a female, say this was a female, down here, the hen, you don't have much feathering. It's just rounded. Rooster feathering can get pretty crazy. Tried to find some nice free images of them. So for a female, it doesn't matter what this female has. This female could be homozygous dominant, heterozygous, or homozygous recessive. No matter what, she's not going to have that pale feathering like a male rooster. So that's sex limitation. Only males, or sometimes females, will show it. All right, next one is called genomic imprinting. You might have heard of this one before, but this is something that typically occurs during developmental stages or fetal stages. So expression of autosomal allele depends on the parent of origin. The two examples here I'm going to talk about. The first one is insulin growth factor 2, or IGF-2. In this case, this is an important gene that is expressed early on in the fetal stages for growth and development. So, a proper expression leads to, large normal size. In this case, the paternal copy is expressed. So if only the paternal copy is expressed, let's say it's, this individual is this heterozygous right here. This is the female one. The female one is dominant, but it doesn't matter. The male one is recessive. So since the male is recessive, this one, the phenotype is almost like IGF-2 isn't being expressed. Whereas typically this heterozygous form should be a normal phenotype. However, because only the paternal copy is expressed, it's related to this. So this is the way this is called genomic imprinting. So expression of the autosomal allele depends on the parent of origin. So same thing here, if the female was the recessive version and the male was dominant, now this male would produce IGF-2 just fine. So, it matters on the parent of origin. And we'll talk about this more later in future chapters because epigenetics could play a role in this too for genomic imprinting. The other example here is a deletion that occurs on chromosome 15. So this one's interesting. So let's say we have chromosome 15 here, and a paternal and maternal copy. So here's the maternal, here's the paternal, and there's a region on chromosome 15. Let's say it's right down here. Now, there's a rare chance event that this region on chromosome 15 could become deleted, which means you lose it. And depending on which parent of origin loses that region, it could develop into a different disorder. So the two different disorders, let's say there's one right here, and the deletion occurred on the male. So we had the deletion right there. The female one still works fine. This becomes something called Prader-Willi syndrome. And the opposite, if that deletion occurs on the female chromosome, and the male one's fine, this one becomes Angelman syndrome. Now, these disorders are sort of similar, but they present slightly different phenotypes in their outcome. So if someone's diagnosed with Prader-Willi or Angelman syndrome, they are slightly different phenotypically. And even though the deletion is in the same part of the chromosome, it matters which parent that chromosomal deletion came from. So kind of interesting there. And then the next one we're going to talk about are genetic maternal effects. So this is determined by the mother's genotype. So all that matters is what the mother passed on. It doesn't matter what the father passed on. So let's say this was the genotype of the offspring, and this was, from the father, and this one was from the mother. This one, the recessive version, is expressed, whether that's good expression or bad expression. It matters what came from the mother. And the main example that's always used here is sinistral or dextral spirals in snails. So right or left. I'll just say right or left. So here, S plus is for, shell spiral or sinistral. S plus is right. And an S is the recessive form and left. So let's say we have an S plus, homozygous S plus male crossed with a left female. So the products of this would be all heterozygous, of course. Now, what should this be? So if I tell you right is the dominant, you would look at this and say, oh, they're all right turns on the shells. However, these are all left. That's because this recessive form here came from the mother. So now let's say we had one of these was the female, so S plus S female here, cross with, let's do an S plus S male. So running out of room, male. So I'm going to write out the Punnett square for this one so we can see it a little better. Okay, so here's that Punnett square, and I added red for female and blue for male here to try to make this a little easier to understand. So when looking at this one, remember, all that matters is what the female gave. So this one, the female gave the S plus, so this one, the female is a right. This one, the female, well, this offspring is a right, not necessarily the female. And then over here, we have an S plus S. This female is left, and this female is left. So here you have an S plus S that's left because all that matters is this one right here that came from the female. So just, wanted to draw that out, make it a little easier to see and understand there. So hopefully that helped. All right. Last one I want to talk about here, it's not necessarily like you don't think of this as being something that's, sex related, but it is cytoplasmic inheritance. So think about it. So, what sort of genes do we have in our cytoplasm? We always talk about genetic material found in the nucleus, or DNA. And that's all we talk about with inheritance. But we do have genes that are found in the cytoplasm as well. So what genes do we have? Or what organelles do we have? The mitochondria, of course. And then plants have an additional organelle that has their own genetic material, the chloroplasts. So remember, at least let's talk about eukaryotes here and, humans, sperm, egg. The mother provides all the mitochondria. The father only provides the genetic information. Nothing else from the sperm enters the egg. So mitochondria then are all from the mother. So organelles with DNA typically inherited only maternally via the ovum for that reason. But I wanted to bring up both mitochondria and chloroplasts here. So some of the shared features so we abbreviate mitochondrial DNA, mtDNA, and cpDNA for chloroplasts. So shared features of each, there are many copies of the chromosome per organelle. So if I were to draw a mitochondria right here, we have the little mitochondrial structure, very horrible mitochondria. There's a whole bunch of circular chromosomes inside that. They also have their own machinery. So you can have variations within the mitochondria of different genes that are found on that. So you can have some variation within the same organelle. Then you can also have variation within the same cell because there are many copies, many versions of that organelle per cell. So lots of different versions in the same cell. And there's some nomenclature we have for this. So if I draw a cell here, and let's say we have a mixture of mitochondria, some are red and some are blue, this is known as homoplasmy. No, said that wrong. I got ahead of myself. This is heteroplasmy. They're different. And then if they are the same, again, the center part here is a horribly drawn nucleus. The same, let's go with all blue ones now. This one is homoplasmy. And your brain gets ahead of your drawings. So, what does all this mean? Remember, I drew it here for mitochondria, but think about it for form of like chloroplasts and plant leaves. So if you have a mutation on chlorophyll, you can have the different, it's called variegated in plants. So you can have leaves of different shades of green and different colors for different forms of chlorophyll found within the chloroplasts. But for something like mitochondrial inherited disorders, they could come from the mother, and those mother eggs could have slight variations within them as well. So you can have this chance of heteroplasmy or homoplasmy in the egg production where you can't be 100% sure on things too. So this is, even though we don't always talk about this when we talk about, our, genes and all this stuff, it's still important to remember that the mitochondria in us do have their own genetic information, multiple copies of that genetic information, and it could vary from cell to cell and within one mitochondria as well. So remember, these are like ancient prokaryotes, and they all divide via binary fission. So kind of cool. In a future chapter, we'll go into more about the endosymbiotic theory, which is really neat to think about. And then right after this, I do have a video it's called Schefflera arboricola. It's a typical plant found in Florida, and I live in Florida, and I went and bought one, and I made a video showing how you can have chloroplasts, variegated chloroplasts in different leaf colors in this plant. And that'll be the next video you see coming out after this one. But that's all I have for today's lectures on sex-related effects, extensions of inheritance. Next video after the chloroplast one, we'll be getting into multi-locus extensions. So I hope you all have a great day, and I'll see you all next time. Bye-bye.